Hi, my name is Dr. Elizabeth Busher, and I'm a senior research scientist here at the Innovation Center with Winfield United. I'm working on new soil assays to look at how much disease pressure you have in a soil sample. Let's go inside the lab and take a look. Soil pathogens, specifically insect and fungal pathogens, cause significant yield loss each year. Sick, diseased, or dying plants divert energy and resources from seed development to plant survival. The yield decrease causes billions of dollars in economic loss each year. Seed treatments, seed varieties, trait selection, planting date, and crop rotation are all excellent management strategies and readily used as holistic management practices. To know which management practices are best, we need to understand pathogen pressure to improve pathogen control, as well as possibly improve yield. To better understand pathogen pressure, a reliable, repeatable soil test would be a powerful tool for Winfield United to provide our owners. Current soil testing focuses on nutrient management. However, we can use that same soil sample to get more information. If we leverage molecular biology, we can use DNA to better understand a field's disease pressure, making a more informed management decision. Currently, no commercial tests are available that target pathogen DNA. What if we could look at the soil pathogens in a soil sample? Winfield United is currently working to do just that. Our first target is soybean sudden death syndrome. Every living thing has DNA. DNA is like a cookbook. It's the instructions that give rise to the cake, or what makes you and me, or what makes a pathogen what it is. It's easier to measure the cookbooks in the soil instead of the cake. So we use molecular biology techniques to take a snapshot of all the soil microbes within a sample. We take a small soil subsample and then we break open those pathogens, release their DNA into a solution, and then we remove all the debris. Now we have clean DNA that we can use to quantify those pathogens using quantitative polymerase chain reaction, or qPCR. Since we're only interested in examining a few pathogens and not all of the DNA-containing entities in the soil, we only need to look at a small piece of DNA for the target. qPCR works like our own cells, which can copy our DNA. We need enough of these copies to properly detect pathogens. As you can see here, we use deoxyribonucleic acid to find the sentence in this large paragraph that we want to copy. Once we start copying this sentence, it becomes easier and easier to copy it. Now, we can compare your soil's disease quantity to known quantity standards. A final report would include your pathogen count and management recommendations based on your unique soil sample. Now you're armed with more information to make more informed management decisions.